everyone, and welcome to Physics 380, Molecular and Cellular Biophysics. My name is Brenda Lee, and I will be your instructor this term for spring 2021. We've been teaching online for about a year now, and hopefully this course will be just as enjoyable for you as it is for me. So the way I've structured this course is pretty similar to what I did last year. I basically have these pre-recorded lecture videos, and I will also be having summaries at the end of each week that are live sessions that will also be recorded. For all of these pre-recorded videos, I have a really specific structure to them. There's a bar at the bottom of each of my slides to tell you where you are. There's also going to be bookmarks for each of the YouTube videos that I post. I usually start with a recap. I go through an introduction of the new topic. I then go through the main content, some examples and applications, and then a quick summary at the end. So these pre-recorded lecture videos are meant to be summaries of a topic. The in-depth discussions will be during the live lectures. So we'll start off with a quick recap. What did we learn from previous courses? For this course, you essentially need to know a little bit of biology and a little bit of physics. But because there are not too many prerequisites, I will be starting from almost scratch for each of these. You will need to know some calculus, and if you have some coding experience, you can have a lot more fun with many topics in this course. But of course, it's not required. So what I'm going to be talking about in this lecture is a few course details, schedule of topics and course objectives, the grade distributions, as well as some assignments and projects. I then will go through an introduction of what molecular and cellular biophysics is, and then wrap it up with a summary. So here's a quick recap, what you should already know a bit about. Some calculus, physics, 112 or 122. You need to know some general physics in order to progress well in this course. And something you should know is some chemistry. So Chem 123, chemical reactions, that is quite important in this course, some biochemistry knowledge. So just to do a quick overview of all of these, you should have already taken Math 127 and 128, which is Calculus 1 and 2 for the sciences. It goes through, obviously, algebraic, logarithmic, and exponential functions, which is very important when we're talking about modeling biophysics. You can take a look at chance and probability, as well as differentiation and integration. So all of these will play a role in allowing us to model different systems in different biological systems using physics. From Physics 111 or 121 and Physics 112 and 122, that's General Physics 1 and 2, the two key things would be simple harmonic motion as well as energy and entropy. Those topics play a key role in being able to understand and create models for biological systems. Last but not least, Chem 123, the chemical reactions that you have learned in that course, such as the ideal gas law, specifically rate equations for chemical kinetics. Those are really important topics to know for Physics 380. So in terms of the course itself, I'm going to walk you through what we will be going through in this term. Of course, you should know who's who, myself and your TAs. I want to go through an outline of the course, what the schedule looks like, and the grading distributions, and what assignments and projects you should look out for. So there are two types of lectures that you should know about. There are the core lectures that are delivered through pre-recorded videos just like this. The slides are going to be on Learn. The live lectures are going to take place every week and will be recorded. Those are really helpful if we have practice problems that we have to go through throughout the term. I'm going to be your instructor. I finished my PhD here under the supervision of Dr. Zoya Leonenko, and I'm really happy to be able to actually teach alongside her now as a faculty member here. So you may have met me before if you've taken Physics 280 or Biology 280. I taught that course in the previous term. I'm very excited to teach this Physics 380 as well. It was my favorite course in undergrad. So how can you reach me? You can reach me through Microsoft Teams. You can also reach me through email. And of course, office hours. This is a fairly small class of approximately 50 people, so I will be available anytime for all of you. If you have a question about any of the concepts, or if you want to learn something more about any of these topics, message me on Microsoft Teams, set up a one-on-one appointment. I'm all for being able to enrich your learning. There are 2 teaching assistants this term. There's Amir and Arena. They're amazing TAs. Their 2 emails are over here, and they'll be monitoring our discussions on Microsoft Teams. Now, keep in mind, I chose Microsoft Teams because there is group work involved in this course, and there will be group work involved in the midterm and final exam. I ran a midterm and final exam with some group work last year, and it worked very, very well. So I'm hoping to have the same experience for all of you this term. So course topics and schedule. We first want to start off with an introduction to molecular and cellular biophysics, and then take a look at how we actually can model something at the molecular level. And in order to model something at the molecular level, we should probably know how these molecules interact with each other. So that's why we have to first go through intermolecular interactions. After that, we're splitting up our discussion into proteins, DNAs, and lipids. So a big portion of the course will be divvied up amongst these 3. Okay. In terms of the grading scheme, your final grade will be calculated using the following scheme. It's 30% assignments. There's a 30% literature review. The reason why I have a literature review here, it's basically an essay, but most importantly, if you are moving on to graduate school, this is one of the first things that you'll have to do if you want to do any research. Learn as much as you can about a topic that you're very interested in, write a report and a summary about it, and then present it to somebody. So in this case, I think it's a really good skill and a really good exercise to do so that you're prepared for a future career in academia. Next, you have a midterm and a final assessment. They're each 20%. They're just called term assessments because it's not officially a midterm or a final exam. It's just similar to a midterm and final exam. So these will involve a multiple choice component as well as a group work component. There's also a 5% bonus that you can choose to complete, which is due at the end of the term on the last day of classes. No exceptions for late submissions. Okay, so first there are weekly assignments that are worth 30% of your final grade. There's a total of 9 assignments, but the lowest one will be dropped, so only 8 of them will count. We're going to be using CrowdMark to submit the assignments. There are no assignments in the first week, so I want that first week to be relaxed for everybody, get familiar with your classmates on Microsoft Teams, and also figure out your schedule for the rest of the term. One thing I do want to mention is that it's really important that you watch the lectures or just read the slideshows because we will be going through practice problems together, and if you do attend the live lectures, I'll try to help as much as possible for each of your weekly assignments. The solutions to the assignments will also be posted after their due dates. Okay, so the next part I want to talk about is the literature review, which is worth 30% of your final grade, and it's a big chunk. So one of the most important things about literature reviews is that you have to pick a topic you're very interested in and read many papers about it. So if I am a newcomer to this topic and I read your literature review, I should get a very thorough introduction to that topic. So because this isn't graduate school and this is undergrad, I only want you to write a condensed literature review. So they're very important in honors projects if you do choose to do a research term, as well as graduate studies. Usually literature reviews are 5 to 50 pages depending on your topic and scope, but the condensed literature review that I want you to write is 10 to 15 pages in length. An entire set of guidelines will be posted on Learn sometime this week, and you can get started right away if you wish, because you have all term to work on it. I will also be creating milestones throughout the whole term so that you can hit them and stay on track. One of the important things to know about the literature review is that you get to pick your topic. So it has to be something you haven't studied before, but it can be anything related to molecular and cellular biophysics. So if you want to learn about something that you haven't learned about before, take this opportunity to try and learn it. If you know that you're going to go into an honors project later on, and you have to do a literature review for that, why not get started right away? So there are several milestones that I talked about. The first one is to pick your topic. That's due May 28th. Use the Dropbox on Learn to submit your chosen topic. Just being able to submit it and choose a good topic is worth 2 marks. The second milestone is to be able to create an outline. That's due June 11th. So you basically have 2 to 3 weeks to work out an outline for your project. Once again, submit it on Dropbox. It's worth 3 marks. And the 3 marks are basically for each of these points below. You have to have a table of contents to let me know what sections you're going to be writing about, just so that I can give you some input and feedback. You have to have a list of at least 10 references that you've found that are suitable to your topic. And you have to write a maximum 300-word abstract that describes your topic to show me that you've actually done a little bit of research and know what you're going to talk about. There's going to be a second set of milestones. I call them milestone 3 and 4 because they're drafts. So you get 2 chances to submit a draft for me to look at. Because these drafts, or because the literature review is so time consuming, I do want to make sure that everybody is on track and also gets as much feedback as possible. The TAs will also be holding tutorials and sessions to help you work with these literature reviews. So the draft is worth 15 marks, and you can choose to submit it on June 25th or July 9th. So June 25th means that if you submit a draft for me and the TAs to look at on the 25th, we will take a look at it and give you feedback and give you a mark out of 15. If you do not get 15 out of 15, you have until July 9th to resubmit your draft and get a better mark. So you have the opportunity to make up for any lost marks in the second checkpoint. If you choose not to submit anything on June 25th and you submit your draft on July 9th, then you will not be getting feedback until after July 9th, and that means you will not be given a chance to resubmit a draft to get your full 15 marks if you don't get perfect. Next is the last 2 milestones, milestone 5 and 6. 5 is basically just submitting it to the Dropbox, turn it in to see if you've plagiarized or not. Keep in mind, I've caught people who've plagiarized before. Please do use the Turnitin software to make sure you're not plagiarizing anything, and to make sure that it goes through all of the checkpoints properly. So milestone number 5 is basically you submitting it to Turnitin to make sure that you meet the minimum threshold for plagiarism. And if it is okay and you're ready to submit, then you can move on to milestone number 6 and just submit it. So in terms of the actual literature review, it's going to be out of 100 marks, but it's only worth 80% of your whole literature review mark because you have the other milestones before. In terms of the rubric, it says the rubric has already been posted on Learn. It will be posted on Learn sometime this week. Another important thing to note is the final version of your literature review is due July 30th at 11:59 p.m. midnight Eastern Standard Time. I will not be accepting any late submissions unless there is a VIF, just because I need the time to mark it all. So what is included in the literature review? The completed literature review, the finished product is to be uploaded on Learn's Dropbox. Needs to have a title page, it needs to have an abstract, no more than 300 words, And the main literature review has to be 10 to 15 pages in length, single spaced on letter size paper. Citations should be numerical and in the order of appearance, such as 1 or 2. This is because there are a lot of students that I've caught in the past who have overused references, especially references with many, many names. So if they appear too many times, it's very hard to get a judge of how much content there actually is versus citations. In order to have a good literature review, it's very important that you use subheadings so that it's very easy for the reader to keep track of where they are. So having that having that table of contents and having subheadings will help significantly. Next, this is a literature review, so I do expect at least 20 references published within the past 5 years, or preferably published within the past 5 years, so that your lit review can be as recent as possible. Now, the last thing I want to talk about is the bonus project. I do have an available bonus worth 5% that's on top of the final grade, and essentially what this is throughout the term I'll be releasing some bonus lectures that talk about how modeling takes place in real life research. So I'll be focusing in on some certain topics that I'm interested in, and you're asked to think about, design, and implement an improved model, one of the models that I present, for a bonus mark. So I'll be presenting a very simplified model for one topic, and if you think that's a bad model, then try and suggest a better one for a 5% bonus. So what do you have to submit? I'm very flexible with how you want to approach it. You can either provide me with a written document, you can provide me with a presentation, or you can even code something up and give me a program script with explanations to showcase what you're thinking about. I will also be posting full details on Learn under a separate bonus folder. Now, because it is a bonus that's on top of the final grade, the deadline for that bonus is August 5th at 11:59 p.m. That's the last day of class, and I'm not allowed to accept any submissions after the last day of class. So, course materials. There are lecture notes and videos as the main resources to use for this course. Please note that the lecture notes do include figures taken from external resources such as textbooks and copyrighted material. I try to draw as many of the diagrams as I can, but some of them are just too complicated, so they are copyrighted from other places, but I don't use too many of those. And because of that, my lectures cannot be reproduced elsewhere or handed out by students, or else there will be Copyright Act invoked. In terms of textbooks, none are required, but there are many, many resources that you can use. I'll show them after. Each textbook is technically catered to a different audience, so depending on whether you're more of a biologist or a physicist or a theorist or an experimentalist, certain textbooks might be preferred for you. So these are the 4 textbooks. I will go through them in a little bit more detail during the live lecture and try to pinpoint which topics to focus on for different books. But for now, if you're interested, you are more than welcome to see if you can get your hands on one of these, especially if you're going to move forward into biophysics in the future. So, in terms of the midterm and the final exam, on June 25th, 2021, there will be a take-home assessment. You have 24 hours to submit the assessment through Learn. The final exam is August 4th to August 5th. It's basically a 48-hour exam. You have time to work in your groups to solve and to model something, and more details to come in the later lectures. Now for the administrative stuff, if you have any missed tests or assignments, you have to submit a VIF in order for me to do anything about it. If you do have a lot of things due in a certain week, let me know. I try to be as flexible as possible on a case-by-case basis. If in general the entire class has a conflict, then I will try and postpone things to make it more convenient for everybody. Some things to mention: academic integrity. Please do not plagiarize, because if I do catch anybody plagiarizing, it will lead to a case of academic offense, and that's always not fun. In terms of viewing assignments and exams, I'll try to mark these assignments and exams within 1 to 2 weeks so that you can view them immediately after. So, is Physics 380 useful? I personally think it really is. It's an extension of Physics 280 or Bio 280, Introduction to Biophysics. It has some overlap with Physics 225, which is Modeling Life Physics, but it's very useful for Physics 395 and 396 if you choose to use it in the future. And of course, it is very useful if you are moving into biophysics research. So with that introduction done with, we can actually move on to an introduction to cellular and molecular biophysics. So what we're going to cover are some definitions. We're going to focus on the molecular level, and we're going to look at some interactions and structures at the molecular level as well, along with what it means to have biological complexity and what a model is. So first, I want to go through the subsets of biophysics. We should probably know the difference between molecular and cellular biophysics before we proceed, just so we know what this course is about. So let's start with biophysics. Biophysics, as you've probably guessed, is the bridge between biology and physics. Biology usually presents a problem that we have to solve, and physics provides the quantitative tools, both abstract and predictive tools, to solve them. So molecular biophysics is a subset of biophysics where biological problems presented are on the molecular scale. Biomolecules and interactions with other molecules will be studied, so that's our focus for this term. Next, you have cellular biophysics. So that's a subset of biophysics focusing on the physical principles used in cell function. So this is an example of kinesin transporting a vesicle. So it's actually using protein dynamics on the nanoscale. So through modeling, we're actually able to create images like this and predict the type of movement that certain molecules will have. So I just like this GIF, it's pretty cool. So moving on, we should talk about the significance of biophysics, because this is probably your first venture into true biophysics. So how does physics play a role in biology? Let's start off by mentioning that when you want quantitative data, you need a quantitative model. And before you even get quantitative data, you have to prove that your model even works, and that your model can produce results that are comparable with tested biological phenomena. So in order to even use a model, you kind of have to suggest one first, test it out to see if it works with with whatever you're studying, compare your results with what actually happens in real life, and see if they're comparable or if they correlate well. If they do, then that means you have a good model and you can proceed with predicting additional interactions and additional results without going back to the real thing. So one of the first steps of building a model is determining which features of the problem are of central importance. Basically, what variables and or parameters do I have to look at, and how are they related to one another? So physicists were actually some of the first ones who invented ways of approaching biological problems that could be simply solved with a microscope or visual observations. Physicists actually tried to bridge the worlds of theory and experiment, and that's where biophysics emerged. And because of that, there are still physicists or biophysicists that try to work on the theory to develop different models for experimentalists like me to attempt to use and compare with real data. So nowadays, this has proven to be very useful because we can use physics to model and analyze really complex systems, or even manipulate single molecules. So it's all still a budding field, with tons of questions that have to be answered and many more discoveries to be made, which is what makes it so awesome. And here I'm showing a picture as an example of discoveries made by physicists and biologists together. So if you don't recognize this, you should. It's an example of the proof of DNA as a double helix. So without physics knowledge and background, this X-ray diffraction image wouldn't even have been possible. So something really interesting about this is this was taken by a PhD student named Raymond Gosling. So, wait, his picture has disappeared. There's his picture. So Raymond Gosling passed away in, I believe, 2015, so just a few years ago. But he was the PhD student who took the first image of the DNA double helix. So it's called photo 51 for the 51st X-ray diffraction image that was taken by himself and his PhD supervisor, Rosalind Franklin. And unfortunately, what happened was Rosalind Franklin wanted to focus on a different form of DNA a little bit more, so she kind of wrote this off for a bit and then asked him to give the image to his old supervisor, Maurice Wilkins. So Maurice Wilkins then showed this picture to James Watson, who immediately recognized it as proof of a double helix or helical structure, and he put two and two together with Francis Crick to discover the structure of DNA. So tiny things can add up to really big discoveries, and in terms of the discovery of the DNA double helical structure, wouldn't have been possible without a physicist behind all of it. So there's a famous quote from Francis Crick in his book trying to summarize his goals and what others should attempt to do. He said that, number one, almost all aspects of life are engineered at the molecular level, meaning all biological function stems from different interactions at the molecular level. He also said, number two, that all approaches at a higher level are suspect until confirmed at the molecular level. So if you're trying to do an approximation at a higher level, they're just approximations until you can confirm the behavior at a lower level so that everything can be explained at each step. So this means we have to understand how and why something works at the smallest level with great detail before we can assume the cause of any higher functions. And that is essentially the goal of molecular and cellular biophysics. So what exactly is molecular and cellular biophysics? First of all, this course focuses a lot more on the molecular side of things. So in this chain, I've ordered things from big to small. So big starting from cellular communities and going down to cells, to molecular structure, to biomolecules, then single molecules, and then atoms. So I've circled biomolecules and molecular structures here because that is our focus for the entire course. So we want to look at the building blocks of these four main macromolecules and see how they self-assemble into their molecular structures. So life in biophysics here takes on a different meaning. Especially in some textbooks, they use life to describe different properties of living organisms that make them living. So the pursuit of understanding the nature of life essentially means that we have to investigate the different properties of living organisms and their constituent materials. So number one, we know all living things are made of cells. So in fact that we know that organisms are constructed from the four great classes of macromolecules. So proteins, nucleic acids, carbohydrates, and lipids. They're pretty different on the molecular or the macromolecular scale. So we call this the diversity of life. However, despite this diversity, if we zoom in a little closer and look at the kinds of atoms found within them all of these diverse macromolecules consist mainly of carbon, oxygen, nitrogen, hydrogen, and some sulfur and phosphorus. So it's mainly the way in which this set of atoms are connected to each other that creates the special properties of the macromolecules found in living systems. Because of this, we call the molecular scale a level of unity in life. So the more you zoom in, the more unified everything becomes, because everything's made up of the same building blocks. But the higher up you go, the more diversity you get, because every step that you go up, the more configurations you can have, and therefore the more functions you can have, and the more differences. So, for example, even within my research, my drug that I studied, not mine, I didn't invent it, the drug I studied, daptomycin, had 13 amino acids, and somehow it didn't work inside lung surfactant. It just got stuck to lung surfactant. But if you changed one of those amino acids, because I also tested other derivatives, if you changed one of those amino acids, it suddenly worked again, but then it became toxic. So just by changing one building block, you can change the functionality of one molecule quite significantly. So throughout this course, we're going to take a closer look at the unity at the molecular level. And cells use the same chemical building blocks to build their biomolecules, such as DNA, protein, etc. And we're going to see how those building blocks interact with each other to give function to each macromolecule. And since we're looking at the molecular level, we can also assume that their physical and mechanical properties are very similar, allowing us to use models to represent their properties. So here's just a brief overview of the 4 great macromolecules. I don't want to go into them into too much detail with them because we'll be focusing on each one throughout the whole course. Over here we have nucleic acids. So they play a crucial role in storing and transmitting genetic information. You also have lipids. So it stores long-term energy and it protects via the formation of barriers and membranes. You have proteins. They do most of the work in the cells and help with cell structure, function, and the regulation of the host. Then you have carbohydrates, which are used for energy, storage, and surface properties. So with carbohydrates, not really going to focus on them as much as the others. In fact, we'll just say we won't really focus on it at all. We'll mainly look at DNA, lipid membranes, and different protein structures. This leads us to the central dogma of molecular biology. So since we're doing mainly molecular biophysics, we should understand what the central dogma of molecular bio is. So this describes the flow of genetic information within a biological system, so from DNA replication to transcription to translation. But the key thing here isn't the process itself, it's the fact that differences or different arrangements of building blocks can create different pieces of information or different ways of expression. So when we look at a model, we have to look at the building blocks and how they're linked together in a chain, and then we're going to have to see what kind of information they carry in order to see the whole picture. So the next few slides are basically just going to go through different ways of modeling different macromolecules. We have DNA here, and we can only model one aspect at a time. So that means we have, for example, we can model it as a sequence. So this is something you're probably familiar with if you've taken molecular bio. You also have binding sites, so you can model it through that. Some of the more physical models for DNA include modeling it as a charged rod because it's so negatively charged. You can actually model it as one rod of negativity. You can also model it as an elastic rod because it does stretch and it does compress, so you can model it as an elastic. And of course, you can model it as a random walk chain, so that's something we'll be looking at as well. After that, you have modeling proteins. So we can model proteins through different ways. We can model it through an amino acid sequence. You can model it through an HP model, and primarily we'll be using the HP model, which means we're looking at hydrophobic or hydrophilic amino acids and sequences. So we can just model it as two things to simplify it. We can look at random walks for native states, and we can also look at alpha helices and beta sheets for secondary structures. For modeling lipid membranes, my favorite, you can model them as different types of surfaces based on phospholipid properties. So you can use an array of springs, you can use a random surface, you can use an RC circuit or parallel plate capacitor. You can also use a model of a semi-permeable membrane where things can kind of go through if they're too big and if they're too small, and if they're too big, they get stuck. So the one thing I haven't mentioned here is whenever you model a lipid membrane, there's so many lipids to choose from and so many different types, you're going to have to simplify it into possibly different lengths of phospholipids or different properties. So anytime you're doing a lipid membrane model, let's say of a bacterial membrane model, every different type of bacteria, especially gram-positive versus gram-negative bacteria, they're going to have different lipid compositions for their membranes. So depending on which bacteria you're looking at and what type it is, you're going to have to use a different set of lipids. Possibly, let's say, if you have a bunch of positively charged lipids, maybe just use one representative positively charged lipid in the exact same ratio for that model. And then you can create your own model to see if it mimics real life. Next, we want to model cell movement. So cell movement can be modeled in different ways. Here we have the idealization of E. coli. So this cell can be modeled as an array of receptors for a ligand of interest. You can also model it as an elasto-hydrodynamic object, so basically the swimmer. You can model it as a random walk that's biased and moving through water. You can also model it as an information processing device characterized by a series of genetic networks. Finally, we can also model solutions. So it can be represented as a lattice of discrete positions where solutes might exist. You can model it as a fluid with some sort of average flow. You can model it as a viscous medium that exerts drag force on anything that goes through it. You can also have a fluctuating environment, which gives rise to random motion in macromolecules. You can model it as a hydrophilic medium that repels, that dissolves polar molecules and repels oil-like molecules. You can also model it as a dielectric medium for electrochemical studies. So for us, we'll be focusing on the latter two over here because our macromolecules are in some fluid at all times. Next, I'm posing a question, basically, can everything be a spring? So obviously not, but in a model they can be. So there are tons of biological models that are based on simple springs in classical mechanics. From here, the assumption is that when a spring is relaxed and at resting length, the energy involved is at a minimum. So right here, when the spring is relaxed, the energy is at a minimum right here. But when it's compressed or when stretched, the energy increases, either kinetically or potentially. And this model can be applied to so many different things. So if you're doing AFM imaging, imagine this is the tip of the AFM cantilever. The more it presses down, the more resistance there's going to be from the surface, which means there's going to be a bend. The more a bend, the more potential energy that's stored in the spring. You can use optical tweezers. Cell membrane fluctuating would involve some sort of energy as well. You even have energy involved in random walk environments. So if you're looking at a DNA polymer that's wiggling around in a solution, you can probably relate its energy to a simple harmonic oscillator. So the key thing here is that we can associate a simple harmonic oscillator to a whole bunch of biological systems. So just as a summary, what did we learn, and an overview of deadlines for the course. Through this lecture, you should have learned some physical tools that allow us to study life on a molecular level. We explained the importance of those tools to study it, and we also looked at different ways that we could use those tools. So when we're talking about modeling, there are so many different ways to look at one biological molecule. Our jobs would be to pick one way to look at it and hope to get some sort of information from it. So molecular biophysics is primarily based on idealizing some sort of biological system, or basically simplifying it into one simple model. And every model and every property can then provide insight into how things work at the molecular level and why higher levels behave a certain way. So I think it's pretty cool that we can merge two fields together and be able to look at something in different perspectives.